Assalamualaikum and good day everyone. So this week we will embark in a new topic which is on principle of proteomics technology and its platform. So unlike genome or genomics, we don't really have that much platform. We don't have different generations like we have generation 2, 2.5, 3 and so forth. So in MS there are a few platforms, but it's basically based on spectrometry or what we call as mass spectrometry. But before that, again, let's have a look on the overview of the proteomics. So we know that samples need to be prepared first. So always, everything starts with a sample. And for sample preparation, basically it depends on the proteome that you want to prepare from. The type of organism, because for bacteria probably it will be easy, and also for animal cells, but for different parts of animal and also different parts of plant, probably you may need to have a very specific method to prepare a sample. So this would be followed by protein separation, either using 1D or 2D. So over here you have a picture of a 2D electrophoresis. If you were to do 2D, you can actually do some spot detection and image analysis. If you were to do 1D, you could just do some image analysis just to determine the thickness or the quantitation of the band. So this would be followed by enzymatic digestion. This is done to chop or cleave your big protein into smaller pieces into peptides, so that the peptide could be detected, and you could basically fingerprint it. So what we call as PMF, not PMS, peptide mass fingerprinting. So this is done via sequencing, via mass spectrometry, and then of course you can do your database search. So the Proteomic toolkit that we're going to do today is for assessing protein-protein interaction, and along the way, for identifying protein, and later on we will have a look at the quantification, and from this topic onwards, we will have a look at some bioinformatic tools for communication for proteomic works. Before we do that, let's have a look at the old method to study or to express or to see how protein is being expressed. So this is done by Western blotting. So I'm sure you have heard of Western blotting. So Western blot is to detect protein. So you have different kinds of blotting, right? How many blotting that you know? So Western is for protein. So when you have southern or when you have northern, or reverse northern, you actually need nucleotide, but Western is for protein. So this technique combine separation power of the SAS page, 1D, together with high recognition specificity of antibody. So to do Western blotting, you need to do 1D gels, not to do never 2D. But you also need to have an antibody. Actually, you need two antibodies. So one antibody will or function against the target protein, which could be later purified from the serum of animals. Usually, the preferred one would be the mice, or sometimes people use rabbits or goats. So they are immunized with this protein, and then you can purify it later on. Or alternatively, if the protein contains some common tag, because nowadays it's quite common to use a tag to make sure that you get your target protein, or sometimes you use an epitope, which is a combination of radioactive. So an antibody against the tag or epitope could be purchased from a commercial source, such as anti-6-his antibody. So in Western blot, again, just to recap, there are 4 steps. So we're not going to go into details. We're just going to take an overview of how this is done. So it starts with the separation of protein using SAS pH. Later on, transfer of proteins onto nitrocellulose membrane for blotting. It's not moving. Never mind. So the third one is for immune reaction. This is whereby it reacts with the antibody that you use, and of course, the last one would be the visualization. So step 1, we know, I have basically covered that in length a few weeks ago. So we will have a look at the second step, which is blotting. So if you remember back your blotting from GE time, I hope you still remember. If not, then hopefully it could jog some of your memory. So blotting for Western blot is also done horizontally or vertically. As you can see here, it is done not horizontally, it is done vertically as well. So that's a special thing about protein. Everything is done vertically. So when it is done in an upright position, so usually it is actually much better. But having said that, the old ways, before we have the machine for this, So this is actually done in the machine. Okay, so before we have the machine, Western blot was also done horizontally, but it's not very efficient transfer. So that's why they develop a machine. Okay, and using or utilizing electrode, okay, the transfer is actually much better. Now, why couldn't we do this for DNA? Okay, try and Google up, okay, the answer. Okay, there's actually a reason why. All right, so basically your blotting, okay, for Western blot is more or less okay similar to the contraption or to the build-up okay when you do blotting for nucleotides. Okay, so you have your membrane okay in the middle, okay, and then in between them okay you need to have of course your gels okay for the transfer to the membrane. You need to have some filter paper and either a plastic and in this case okay because you are using a machine okay so you have to have plastic. So in the old ways we have glass plates okay just to weigh down so that the transfer could be better. So the third and the fourth step, okay, is of course we know third is detection. Okay, the fourth is visualization, but the third and fourth step usually comes hand in hand, and because detection of it is done and visualization okay is done okay via colorimetric, so you can actually see it okay simultaneously. So this is how we do it. Okay, so imagine just imagine okay because this is a 2D representation. So this is okay your membrane okay where your protein okay have been transferred on it. So when you incubate your membrane okay, with the primary antibody, so the primary antibody will only bind to the protein that is specific to it. Okay, remember affinity. So this is lock and key theory. So the second antibody okay, is the one that is most specific, okay, which will only react okay, to the first antibody. Okay. So what happens okay, is that okay, if you have or if you use colorimetric, okay, you can actually see okay, that the band will light up. Okay. Or if not, then probably if you use radioactive epitope, you can actually just develop the film and then you can see it okay when once you have developed it. So we would know okay that okay which protein okay later on okay would react okay or would be expressed depending on how it binds to the specific antibody, the second antibody. Okay, so this is basically okay, the different kinds of detection that you could use okay either in an enzyme whereby you could have enzyme of fluorophore or colloidal gold okay whereby you could actually visualize it okay using colorimetric or radioisotope whereby you need to develop a film. So that is all about Western blot. Like I said, we're not going to dwell too much into it. Okay, so we're going to take a look at another method. Okay, down memory lane. Okay, yeast to hybrid. Ever heard of yeast to hybrid? If you haven't, I'm not sure whether you cover it in GE or not, or Dr. Yat did mention it somewhere somehow. So yeast to hybrid is another common method to detect protein, but this is more towards protein-protein interaction as compared to expression of protein in Western blot. So in two hybrid system, as the name says, two hybrid, okay, so basically okay, you need to basically have two things over here because this is based on transcription factor whereby it is split into two domains and two hybrid proteins are designed. So one protein okay is of course okay your interest protein or protein of interest which act as a bait. Okay, so they are typically fused to a DNA binding domain. So the proteins will then be screened for interaction with the bait okay to prey, and they are fused okay as a transcription activating domain via transcription activating domain. So as you can see over here, oh, so the writing is actually out of that. Okay, never mind. So the interaction okay between the bait okay and the prey okay will bring these two domains close together okay, which in turn will result in the transcription of a reporter gene. So you get your mRNA to be transcribed. So the reporter gene okay usually are certain essential genes okay or from the protein okay which in case the colony sometimes dies okay if there is no interaction okay or reversely okay the reporter gene can also be attached okay to a green fluorescent protein, but that is harder to be done though it is possible nowadays. So that is the basic principle of yeast to hybrid system. Actually for the yeast system there are yeast 1 hybrid, yeast 2 hybrid and yeast 3 hybrid. So the most common that we use is yeast 2 hybrid. So yeast 2 hybrid is actually quite, I would say typically quite hard to do because I basically spent when I was working in MPOB I spent like around 1 year working on yeast 2 hybrid okay, but the result is not that good. So well, that was in the early 2000s, so now they have a better system. But used to hybrid, okay, once okay you get the result, okay, or if you did everything correctly, okay, and everything is nice, okay, so you could actually see the interaction, because okay, we know that this is based on interaction between the bait and the prey. So if there is no interaction, we know that no reported gene, okay, will be produced, so you will not get your gene. So if there is, then you will see that there is an increased transcription, okay, or abundance, okay, of the reported gene, okay, because of the interaction between the bait and also the prey protein. So in the experimental setup, okay, and because you are dealing with yeast, you also have to bear in mind that you're going to be smelling some not so good smell. So the yeast of the smell of a yeast when you make bread or whatever, okay, it's nice, okay, but yeast when you do used to hybrid, okay, believe me, it's not really that nice. But you have to bear, okay, with the smell of a yeast, okay, during those time. So even though in the diagram, okay, it looks as such, and this is what we do because this involves genetic engineering, but in the experimental setup, okay, what you do, okay, is that you have a petri dish, okay, whereby you have all this bait and the prey protein, and you mix them up, okay, in a solution, and then, okay, you basically put it out on the plate, okay, and then you need to press it, okay, on a velvet cloth, okay, with a special kind of, it's not like your normal hockey stick, okay, it's kind of like something that would press it down, okay, so that means that it should be able, okay, to make sure that those two proteins interact with one another. That is just the protocol. So after about 2 to 3 days, okay, I actually couldn't remember the temperature that we used, but I think it's a bit higher. So we 30 or 40, I actually couldn't remember. So then, okay, we will see, okay, whether there are some yeast, okay, being produced. So the yeast usually, okay, will be extracted, okay, and then you purify the plasmids, okay, and then you do your PCR. If there is any bands being produced, then that means that the reporter gene is being uh, there is an interaction between the two proteins and producing the reporter gene. If there is nothing in your PCR, meaning that your yeast to hybrid actually doesn't work, okay, and you may need to tweak something along the way. So more or less, okay, this is how it is done. All right, so moving on, okay, so those two things, okay, are what people did in the past, okay, people still do it, okay, occasionally if it fits, okay, and for certain kinds of experimental and depending on the type of targets of their research. But from this moment on, we're going to be taking a look at technologies for identifying protein. So the technologies, okay, will involve, okay, protein sequencing, okay, so Western blot, okay, could also be done to not really to identify the protein, but more towards, okay, a target protein, okay, to identify its expression. That's it. Okay, but okay, usually for protein identification, it is by sequencing of the protein. So protein could be sequenced, even though you may have only heard of DNA sequencing. Okay, but protein could be sequenced. So previously, in the old ways, protein was sequenced using chemical sequencing called Edman sequencing. Okay, nowadays, okay, proteins are being sequenced using mass spectrometry. So mass spectrometry is also what we basically do, okay, when we want to identify peptide in mass. Okay, so we call it as peptide mass fingerprint, kind of like your normal DNA fingerprinting. Okay, so when we are using mass spectrometry as compared to the Edman sequencing, okay, you need to do some database searching. You need to do some de novo sequencing if you want to make it a better search and a better result. And also, okay, you need to identify databases and search engines, the suitable ones to be used, and not to be duped, okay, by certain databases that may have kind of like a very dubious kind of input. So let's have a look, okay, at the always. Okay, we always start with always because without the always we wouldn't have the new ones. Okay, so the Edman degradation. So in the Edman degradation, remember this is a chemical sequencing. It's more or less similar to the Sanger sequencing previously, okay, but for protein. So there are only 3 steps. Okay, so the first step, the peptide is treated with phenyl isothiocyanate to give a PTC derivative of phenyl thiocarbamyl. So this is basically a chemical reaction, and because I have never done this, so I actually couldn't walk you through, okay, how it is done in a lab. So step number two, okay, is actually on the reaction, okay, with hydrogen chloride in the anhydrous solution. So the thiocarbonyl sulfur or the PTC derivative attacks the carbonyl carbon, okay, which is basically here, oh sorry, here, okay, and then, okay, what happens is that the N-terminal amino acid, okay, is going to be cleaved. Okay, so this one, okay, will be cleaved. Okay, will be chopped off. Okay, because okay, the thiazole, okay, derivative, okay, will form the remainder, okay, of the peptide. Okay, so this, okay, will be cut off, okay, and then, okay, it will be a separate peptide. Okay, so you have a thiazolone, okay, plus remainder of the peptide. And the third step, okay, so once, okay, these two are formed, the thiazolone derivative isomerizes, okay, to a more stable PTH or phenylthiodentoin derivative, which is isolated and characterized, okay, and okay, you provide the identification of the N-terminal amino acid. So the remainder of the peptide which is formed, okay, in this step 2, okay, can be isolated and subjected to second admin degradation. You do the same thing over and over again. So it's actually a very lengthy procedure and not very accurate at that one. So that's why, okay, we have mass spectrometry nowadays. So how is mass spectrometry different from Edman sequencing? It's actually very different. So have a look, okay, over here. So you have a protein complex whereby when you don't have a specific type of protein, when you just extract protein, it is actually in a crude form. So you have a jumble up, okay, of a mixture of protein. So you can either immunoprecipitate using antibody, okay, or HISTAG and so forth, okay. But okay, a lot of times, okay, people don't really go through this, okay. We just use an enzyme protease, okay, to digest the protein to get a peptide, okay, in a mix, okay, and then okay, we subject to the LCMS. So this is basically the shotgun identification. So you don't think only shotgun only refers to the genome, right? So in protein we also have a shotgun identification. But apart from just the normal shotgun identification, we also have, okay, some very specific and of course expensive form of mass spectrometry, okay, whereby, okay, either from immunoprecipitation, okay, or from that. If you don't do immunoprecipitation, you can just from here to here as well, okay. You just run it on your 1D gel from your SCS page, okay. You just proceed with multi-TOF, or you can just proceed with 2D, okay, either 1D or 2D, and proceed with multi-TOF. So this would be very specific and, like I said, very expensive, because multi-TOF you use a specialized plate and all the chemicals are expensive. So let's have a look at peptide mass fingerprint. What is it? Okay, so remember it's not PMS. So probably the guys who didn't know, you may not get the joke, okay, but this is PMF. So you need to isolate your protein of interest, or probably your protein of interest is among the crude extract protein that you isolate. Never mind, okay, but first and foremost, you need to isolate your protein. Then you cleave or you digest the protein at specific residues. So the term being used, okay, to cut or digest protein, okay, in protein world is called cleavage, okay, and not the other cleavage, okay, but the cleavage, okay, to cut. So you can either proceed with normal, again, mass spectrometry such as the LC, liquid chromatography, or you can use multi-TOF, okay, of the resultant peptides. Either way, okay, using multi or just normal MS, okay, you can extract the peptide masses from the spectrum, so you get lots of spectra, and you search the databases for matching peptide mass fingerprint and determine the peptide and the protein that it corresponds to. So let's have a look at the cleavage, because cleavage is actually very important, okay, in peptide mass fingerprint and protein identification. So cleavage is done using proteases, not just any enzyme. It has to be enzyme from the protease family. So the most common is trypsin, chymotrypsin, okay, pepsin, okay, elastin, and so forth. So trypsin is positively charged amino acid at the C terminus of the peptide, okay, which is the most commonly used protease. And because a lot of machines, okay, either the MALDI or the LCMS, okay, they usually have a standard for trypsin. So, okay, if you use MALDI, ionization, okay, usually shows bias for charged peptides. Hence, okay, it will be suitable for trypsin and chymotrypsin. Apart from using proteases, you could also use chemicals such as cyanide bromide, okay, and some other. But cyanide bromide, okay, is the one that is the most famous. And as you can see, it's actually very, anything with bromide is quite dangerous. Hence, the least favorable, okay, to use to cleave amino acid or protein. And the function or the goal of cleavage or digestion is to chop your large protein into smaller peptides, okay. But other cleavage goals, okay, include experimental proteases or different experimental proteases, okay, to get varieties of peptides. Okay, you could also do some domain mapping from there and also do some conformational mapping, which is actually quite tedious to do. So let's have a look, okay, at some of the proteases. So we're going to take a look at the proteases from the serine proteases family. So they cut at different amino acids. So let's have a look, okay, at these four common serine proteases that has been used to cut or to cleave amino acids. So they are trypsin, okay, that cut specifically at arginine and lysine, okay, chymotrypsin, okay, at basically phenylalanine, tryptophan. I actually could remember what Y is, okay, valine and leucine, okay, but we will come back to that. Okay. Pepsin, F, right, W, Y, and M, methionine, okay, and elastase, okay, basically at A, G, and S. So they basically have very specific amino acids that they cut at. So meaning that you could actually get a very good detection later on. So that's why, okay, among the four, okay, usually trypsin, chymotrypsin, and pepsin are the most commonly used because they share certain similarities in terms of the amino acid that they detect. So cleavage is actually, well, as the name says, you just digest, but it's not as easily digested as in DNA or RNA digestion. So, okay, there are certain issues that people encounter, even I did. Okay, one of them, okay, is contaminating proteins or buffers or deletion, because you may think that you have got a cleaved or digested protein, but it's not, and you may be able, when you run the gel, you can see that some of the proteins, okay, are still visible at a higher range or higher molecular weight. Apart from that, sometimes, okay, because of protein variants, and remember, proteins, okay, have some of the PTM, okay, due to post-translation modification. So the modifications, the splice, and the processing sometimes can result in this variant. Hence, they may not cut efficiently. Apart from that, the types of enzymes being used, okay, so the different types of enzymes, okay, shown in the previous slide, okay, sometimes could cause an autolysis of the fragments or jeopardize the quality. Apart from that, non-specific cleavage is also common. Non-specific meaning that, okay, sometimes it bypasses the first cut site, okay, and only cut at the second cut site, or sometimes it cut prematurely before the cutting site. So non-cleavage, non-specific cleavage. Apart from that, okay, this is, okay, if you think that everything is okay, but then you proceed with MS, then you could see that in MS quality, okay, is actually not as expected. Okay, this could be to the adducts, peak detection, charge state, monoisotopic masses, overlapping isotopes, and many more. Apart from that, if you use chemical, you don't want to use enzyme for any reason, okay, there is a problem with specificities because enzyme basically doesn't cut depending on the amino acid. Okay, so there is also inefficiencies, and because of the modification, it makes it all the more tedious. And the last one would be the database error, okay, after MS, okay, which has a high impact. So that's why, okay, type of database that you choose, okay, to search for your peptide later on matters. Alright, so cleavage fingerprinting, okay, is done to establish protein identity. So take a look, okay, at this example. Okay, so this is a protein. So I'm not going to say, okay, this protein is from where, but this is actually from animal cells. Okay, this is from a journal. So it has been digested or cleaved with chymotrypsin, one of the serine proteases. So have a look, okay, at this is actually the control, okay, and this is basically the variant. So have a look, okay, at chymotrypsin, again, when used at different concentration. So this is the undigested protein. This is how it looks like, okay, 1D. So you can see the pattern. At 75 microgram per liter, okay, so let's just have a look at the control. You can see that lots of proteins are still there, meaning that it is not digested. Okay? Even up until 300 microgram, okay, per meal, which is actually not that much. So you can still see some proteins, okay, but okay, you can see that over here you don't really see that much bands, right? But now, okay, you can see a lot more bands down here. Okay, usually after you have cleaved your protein, when you run the gel, you don't want to see all these bands up above. Okay, you want to see kind of like a dark kind of masses or bands down here, or don't. Usually no bands is also quite good, okay, meaning that it has been cleaved quite efficiently, meaning that it's too small, okay, to be visible on your gel. But it could also mean another thing, meaning that probably it would have been degraded everything. Okay, so that's why, okay, the concentration and also the amount, okay, of enzyme versus the concentration of the protein matters for fingerprinting. And this would later on result or affect the quality of the MS peaks or the spectra that you see. So from the MS you will get the result in peaks, and of course this will be decoded or analyzed further by a software that will tell you each peak corresponds to which amino acid. So it depends on the type of experiment that you want to do. So one of them is that one peak corresponds to one amino acids. Another one is that one peak corresponds to one peptide or one protein. So there are two ways of analyzing it, which we will see. So I will basically just more or less leave you with this diagram, or this picture from a journal in 2009, 2010, I actually don't remember. So the difference is when you cleave the same protein with trypsin versus chymotrypsin, and we know that trypsin and chymotrypsin basically they come from the same family. It's just that chymotrypsin cuts at three more extra amino acids, and this is how they differ in terms of the time, and also the specificity of the cleavage. So different amino acids will be cut differently depending on the kind of enzymes that you use, and probably apart from the concentration, the time that it is exposed to. But you also have to bear in mind a lot of the serine proteases only have a shelf life of about 16 hours. That's it. So more than that, it's nothing. It wouldn't be active anymore. So that would be it for the first video. Next we will move on to the mass spectrometry.